All right, so once again, very basic, and I'm hoping that we could wrap this up in 60 minutes. So why should we care about the liver? And this is a little bit of data that came out from the United Kingdom. Because this 2014 Lancet, so very high-profile journal. And what you can see is that if you look at all the different diseases that produces a lot of mortality and serious illness in humans, and if you track it for the last 30 years or so, 40 years here actually, and I have been seeing this graph for a while, so sometimes time scale gets a little muddled. What you can see is that all these different life-threatening diseases we have paid a lot of attention to, and you can start seeing a payoff here. For example, if you look at something like cancer or metabolic disease or diabetes, a lot of these things are starting to come down or plateau. The number of incidences you see and the mortality that one can see from these diseases. But then, if you look at what's happening in the liver and liver diseases, it's really an astoundingly scary picture. So this summer, I was in Poland and Czech Republic. And in Poland, doesn't matter how small a town you go to, they have an alcohol shop that's open 24 hours a day. So in East Europe, alcoholic liver disease is exploding, and it's exploding in Western Europe too. And you couple that to obesity and some of these other things that's happening, metabolic syndrome, and you have the recipe for like complete disaster here. So this is why it's important that you once again start studying the liver, start studying what's going on, and try to figure out ways we can mitigate this health crisis that is on our hands. All right, so let's take a look at the liver. So if you ask people where's the liver, most people will show you sort of your stomach area, and that's not correct. The liver is actually tucked in under your rib cage, okay? So this is the largest solid organ in the body, depending on if you're a male or a female, if you're fat or small or your height. It's between 1.2 to 1.8 kilograms. So it's pretty big. It is the chemical factory of the body. So the textbook, Alina asked me, what textbook should I recommend? The textbook that I follow has about 1,500 pages. So that's probably not the best thing. And it does so many different things, many biochemical processes, and we'll talk about that. And as I said, it's stuck sort of under your rib cage on the right side. Okay, and this is only typical for the humans. When you go to rats and mice, it's a little bit different, I'll tell you that. And the rounded top surface actually sits under the diaphragm, and the sharper bottom areas protrude out right over here. Now, young kids have a bigger liver. So you may have a nephew or a niece, and they stand, and the liver kind of, like belly kind of protrudes out, and mom says, oh, you're having too much candy. Well, that's not true. And the reason is they have a bigger liver relative to their body, and their upper abdomen kind of like sticks out, okay? And the liver is somewhat protected by your ribs and the thoracic cage, and there's a reason behind it, okay? So any boxing fans here? No? Mixed martial arts? Anybody know who Oscar De La Hoya is? Yes? So Oscar De La Hoya had a fight with this guy, his name was Bernard Hopkins, who was also a very great boxer. And everybody says Oscar De La Hoya threw that fight, and Bernard Hopkins turns out to be his business partner now. So he threw that fight to cement his relationship. But what happened in that fight is that Hopkins actually hit him on the liver, okay? And so it's called a liver blow. And mixed martial artists do that all the time too. That they actually try to hit the liver, they try to go right under the rib. And what happens is it shuts down the blood supply and shuts you down. And so you fall down to the ground, and for a minute or two, you're essentially paralyzed. So I wanted to show a video of it, and then I thought maybe, Friday morning, 10:30, not the best time to do it, but if you guys, want to check it out and want to find out about the liver punch or the liver shot, look it up. It's pretty awesome. Okay.
We
so,
do have Wi-Fi here if you want to try it.
And throughout the lecture, by the way, I'll have some links here, and you guys can go back and look at those links. You'll find more information. I had to pull from many sources on the internet because, as I said, the textbook was grossly inadequate. All right, so liver is one of the first organs that develops in the fetus from the endoderm. And there, its role mainly is hematopoiesis, so manufacturing of blood cells. All right, so let's start with a high-level architecture of liver. And this is really the only slide that I drew. And then it took so much time, I was like, I'm not doing this. It's going to take me like a year. And if I did this every semester, maybe there was a point to it. But right now, it was too difficult. OK, so I use a software called Motofolio. So if you guys have that, you can find little sketches like this that you can put together. It's pretty fun. It's like putting together Lego blocks. Okay, so you have the liver, and I'll talk about the lobes of the liver, and then you have the ligament that holds the liver in its place. And then really the feature I want you to focus on the most is the gallbladder, which is very critical. And what you see is that the gallbladder is fed upon by a lot of these branches. They're called the hepatic duct. Okay? And the hepatic duct, what it does is, one of the main functions of the liver is to make bile. And you probably have all heard of bile, right? And then the bile travels through the hepatic duct, and you can see there are branches of the hepatic duct all over the liver. And they then start draining into the cystic duct, and it goes into the gallbladder. And the gallbladder stores bile, okay? And this is a closed system, okay? So when, under normal physiological condition, when there's not a need for bile, you can see that this architecture then becomes the common bile duct, and it drains down here. And there, it is connected to the pancreas and the pancreatic duct. So your pancreas makes other enzymes, pancreatic juices, okay? And this is a closed system, and there's a sphincter here called the sphincter of Oddi, okay, O-D-D-I, which keeps this thing closed. And this is where the two sort of the gallbladder duct and the pancreatic ducts come together, and this is called an ampulla of Weyner. So this is a closed system, okay? So under normal physiological condition, when bile is not needed, it's closed. And then it all drains into the upper part of the duodenum, okay? And so what happens is when there's a physiological signal, and this is something you guys can look up, and we'll talk about that a little bit, the gallbladder gets a signal from the body, and we'll talk about that a little bit later, and it's regulated by a hormone called cholecystokinin, okay, CSK. And you guys can, I think I have cholecystokinin and some of those other things in a slide later on. And then what happens is there's a squeezing action, so almost like the gallbladder squeezes. And at that point, the bile comes out, it travels through the common bile duct, okay? And the sphincter opens up, and then you have this ampulla, and all of this is then released into your duodenum. Everybody got that? All right. So this is the high-level architecture of the liver. This is one of the main functions of the liver, and it's important we understand this. All right. Hepatic blood flow. This is also another important physiological consideration regarding the liver that you must understand. So after I put this up, and I have words like inferior vena cava, aorta, arteries, veins, I was like, do you ever remember cardiac biology? Do you guys remember how the heart functions? What the superior and the inferior vena cava is? Yes? No? Maybe? Okay, so everybody's nodding their head, so I'm assuming I don't have to do like that. So I am a member of the cardiovascular section here, although I'm not a cardiologist, so I could do a 5 or 10 minute, I could draw a heart and show you how it works. No? Okay, if you have time. All right, so the liver is very interesting when it comes to its blood supply, and it's important because the liver is so metabolically active, it's critical, and it receives a good supply of blood, all right? And this is essentially the piping of the liver that controls blood supply. Okay, so we're gonna start, so we're gonna start with the heart. And the right side of the heart is usually pumping out, the left side of the heart, okay, this is the convention here, right? So this is left and this is right. Okay, this is not left, so it's almost like this is your left side, this is your right side. So the left side of the heart pumps blood out through your aorta. And then the aorta comes out of the main aorta and goes through your body to the abdominal aorta and comes to the hepatic artery. And then the hepatic artery then sends blood along to the liver. Okay? This is the biggest source of oxygenated blood in the liver. Okay? And there's also another source of blood which comes from the hepatic portal vein. So when we talk about the liver, we take out this hepatic part and we just say portal vein. And there's a reason behind that. Okay? You also have the hepatic veins, and we'll get to that in a second. All right? The hepatic portal vein is fed by many parts of the GI tract. So once food gets into your stomach and starts getting digested, the hepatic vein starts getting its supply of mostly deoxygenated blood from all these other veins. So the mesenteric vein and the splenic vein. And you can see it says here it comes from different portions of the stomach, the pancreas, and portions of the large intestine. Okay? And the superior mesenteric vein, in turn, also gets its supply from small intestines, parts of the large intestine, stomach, and the pancreas. Okay, and all of these combines and tributaries to become the hepatic portal vein. Now, the portal vein Okay, so the hepatic artery has two branches. One is called the right hepatic artery, and then the other one is called the left hepatic artery. And if you have to guess, the right artery goes into the right side of the liver, and the left goes to the left side of the liver. Okay? And then the hepatic portal vein. The hepatic portal vein is unique in that it brings blood into the liver that's mostly deoxygenated. Okay? And the convention used in physiology is this deoxygenated blood. It's blue. It's oxygenated blood. It's red. All right? But there's no such thing as completely deoxygenated blood, right? So that's a misnomer. So when people say, well, blood is deoxygenated, well, it's only partially deoxygenated. So about 70% of the blood that you see is so the blood has lost about 70% of its oxygen content. So it still has some oxygenation. Otherwise, when you think about it, if the right side of the heart or any organ really doesn't have any oxygenation, then those organs won't survive. So it's a misnomer. So we talk about it, nobody explains why. I was like, oh, doesn't make sense. But anyway. And then the aorta and the hepatic artery, the left and right side of it, and then that branches off into many small branches, brings in oxygenated blood. And the cool thing about the liver is these two blood streams actually mix together. Now the liver has a really high metabolic capacity, so there's a mixing of both sources of liver. I'll show you a picture of that later on. And so the liver then utilizes this blood to do biotransformation, metabolism, production of all these different factors, and then it's drained by the hepatic veins, okay? And so there's some confusion here, and I said there's a reason we don't say hepatic portal veins is because people confuse these two. All right? So remember, hepatic veins carry blood out and puts it into the interior vena cava, and the hepatic portal veins are just portal veins. It's easier to remember it that way. Brings nutrient-rich blood into your liver. And then this hepatic vein has three main branches. You may want to write this down. It's called left, right, and central. Okay? So one thing that you may want to consider when you look at the liver is that where do you think most of our bacteria in the body lives? What part of our body do you think gets most exposure to bacteria and foreign objects? Yes, yes. So the liver has to be extra vigilant when it handles blood. Because look at this, it's coming straight from our gut and the stomach. So anything that we ingest that's full of bacteria, the liver sees it first, okay? And this phenomena, what that requires is that the liver has a very active immune system. And it does have a very active immune system. We'll talk about that, okay? It's called Kupffer cells. They are the resident macrophages of the liver. Okay, and the liver has a big function. One of the biggest functions, and you'll learn about this in later lectures, is that detoxification is a big function of the liver. All right? Now, one thing that you will hear throughout this lecture is first-pass metabolism. What does that mean? First-pass metabolism is when you ingest something, right? It goes through your stomach, and the first thing it sees where it gets chewed up, well, okay, so I'll say it, but that's not necessarily true, but most of the majority of biotransformation of drugs, foods, everything else, that goes through the liver is done by the liver. So that's first-pass metabolism, okay? So when you give a drug to a person, many of you want to be doctors, you will notice that if it's a drug that's given orally, it's given at a much higher dose than if it's given IV, all right? Because when you give IV, you're gonna bypass this first-pass metabolism phenomenon that you see with oral dosing of drug. So as much as 70% of a drug is broken down by the time it leaves liver. All right? So this is taken into account. And so you will hear terms like clearance, which is how quickly the body gets rid of a drug. Other factors are considered too, such as half-life of a drug, which tells you how long, what is the numerical half-life of a drug in your body. And you will also hear about AUC, or area under the curve, or how much of the drug is present in the body at a given time. But remember, the liver is probably the key regulator of whether or not the organs that you target with your drug will get to see the drug or not. So remember that. All right, so lobes of the liver. This is somewhat important that you understand this. And once again, I said focus on the fundamentals. All right, so if you open up my chest and look at my liver, which will be over here, you will see the two bigger lobes of the liver. So we'll start over here. So right lobe, once again the convention is, this is my left, this is my right. Right lobe and the left lobe, and then you have this ligaments. And there are many ligaments. There's the coronary ligament, there's the falciform ligament, and there's the round ligament. And really, the main function of the ligament is to attach the liver onto your onto the body. And what you will also see is that the gallbladder is protruding out. And then if you flip the liver over, you will see there's the left lobe and the right lobe, and then there's two other lobes. One is called the caudate lobe, which is right over here, and then you will see the quadrant lobe, which is right here. And then you see the architecture of the liver and the piping of the liver right over here. Okay? And you see the gallbladder tucked right in between, and you can see part of the gallbladder veins kind of joined here. The liver is also sculptured. So if you were to take out the liver of the body, you will see the renal impression, which is down over here. This is where your kidneys kind of press down on the liver, so the liver has to grow around it. And then the gastric impression, which is over here, which is due to the gastric in the stomach. Right. Another thing that people don't talk about here, and this is recently, this is eye review grants, and this is something people are starting to focus on, is the Glisson's capsule. The Glisson's capsule is a layer of cells, connective tissue cells, that's fibrous, and it covers the top surface of the liver. Okay, it's about 200 microns in humans, and then you have the liver parenchyma over here. So the liver is covered with the Glisson's capsule. All right. So anybody thinking about graduate school? PhDs? Yes? Okay. All right. So great. So in graduate school, you're doing fundamental research, and you're working mostly with mice and rats, maybe pigs, maybe monkeys. Wait for it, doesn't monkeys. So how is the liver of the rodents different? Okay. And so rodent livers have 4 lobes. Oops, sorry. So you can see over here, you have the median lobe, which really is flat. Okay. So it looks like a flat, and it's got 2 sort of big big sort of left and right side. Then you have the lateral lobe, not the lateral lobe, the median lobe, the left lateral lobe, and the right lateral lobe. And then you also have the caudate lobe, but what you don't have is the quadrant lobe. Now, this is something, if you really want to be mean, get an unsuspecting graduate student, give them a rat, and tell them to find the gallbladder. Okay. Rats do not have a gallbladder. If they find the gallbladder, because rats are one of those organisms, along with horses, they secrete their bile already concentrated. So their mechanism of bile storage and concentration is not required. Okay? All right. And I found this website while I was doing some work trying to put this lecture together. This actually takes you through a rat dissection. So if you're ever curious and want to spend some time really looking at internal organs of a rat, you can go to this website and take you step by step by step. So it's really good. All right, liver lobules. So the liver lobule is a functional unit of the liver. It's also called the acinus, and each liver has approximately 100,000 liver lobules. Okay, and this was described in 1665, so a long time ago. And the reason is the classical liver lobule is hexagonal or polygonal in structure, and 2 millimeter by 1 millimeter. So 2 millimeter by 1 millimeter is very small, but if you have something like a magnifying glass or a reasonably good microscope, you might be able to tell it. Okay? And then it is separated by intralobular connective tissue. And then the corners are marked by what is called portal triads. Okay? So portal triads are the areas of the liver where you have a branch of the hepatic artery, the portal vein, not the hepatic vein, the portal vein, and the bile that all comes together. Okay? Now the hepatic artery, so let's take a look at the lobule. So here's your lobule, okay? So in the corners, you can see the portal areas, and this is from a pig, by the way. In the center, you have the central veins, all right? And I will teach you a way by which you can distinguish between the two. And then you have this connective tissue. See this? Okay? So now, if you take a closer look, and this is a microscopic view at a higher magnification of one of these portal areas, you can see portal veins. Here's your hepatic artery. Okay? how do you tell your arteries apart from your veins? So cardiovascular knowledge test, quick.
The wall. The wall of the arteries thicker than.
Yes, yes. Because the left side of your heart is pumping blood, right? So those are your arteries. They have to have thicker wall. Okay? Veins, on the other hand, are draining blood. So veins have thinner walls. Also, if the volume is constant, and your artery has to handle 5 gallons of blood, let's just say, and your veins have to handle 5 gallons of blood, think about it, then the veins have to be wider, right? Because it has to pass the same amount of fluid through. All right? And then here you have your bile duct. And the way to tell a bile duct is it's usually, there's this hole and then it's surrounded by cuboidal epithelial cells that stain really nicely. And what you see around here is fibrous tissue. Okay, so it's the same fibrous tissue that you see over here. Somebody got that? Okay, so you have your portal veins, your bile duct, and your artery. So pathologists, by the way, those who are thinking about a career in medicine, they are the highest paid people. And not unlike surgeon, you can be a pathologist in your 90 as long as your eyesight is good. Okay? Because this is a perfect slide. In reality, when you start looking at slides and you're a pathologist, it's very hard. You have to go through hundreds of thousands of slides to be good. And so it's important when you guys become doctors that you understand what histology looks like. Pay a lot of attention to histology. All right, so liver lobules, how are we doing on time? Okay. So this is really a cartoon of the liver lobule, and there's a lot of information here. Okay, so one thing that you want to pay attention to is that the hepatocytes are bilayers of cells. Okay, so they're always in sort of these bilayers, and they're polarized. All right. So what you see, let's start from the top here. So what I showed you in the corner was the portal triad, which you also see over here. And then on the other side, you have the central vein. So remember the architecture. So here's your central vein, and here's your portal triad. So we're looking at the same thing. All right? Now, you have these small sort of ducts, which are portal venules. So small veins, venules, right? And then you also have bile ductules, which are basically really small bile ducts. Okay? And what you see here is one thing that you pay attention to is that the bile and the blood never really mix in the liver. Okay? Look at the direction of the blood flow. It goes out from your portal triad into your central vein. Whereas the bile actually flows from the hepatocytes out into the bile ductule. The two don't mix. So anybody want to guess? You guys have probably gone through the slides, haven't you? What happens when these two mix? There's a physiological transition. If somebody has liver disease, say,
Cirrhosis? Cirrhosis? No.
you can tell by looking at their skin. What happens?
Jaundice.
Yes. That's what happens, right? Okay. Now, these are the hepatocytes, and the hepatocytes, the apical side of the hepatocytes, basically join together in this bilayer shape, and from the apical side, you see bile flow. Now, if you open up a slice of liver, it's very hard to see this bile ductules. Under electron micrograms, you can, but if you just take a slice and look at it, these are very, very hard to see. Now, there are quite a few other features that I would like to point out. The first thing I want to point out is sort of this little, cells that are over here. See this? These are called stellate cells. And these cells were sort of ignored up until, I would say, the mid-80s, maybe the 90s. And this cell provides structural support to the liver, and it's also the main cell that is activated when you get fibrosis of the liver. And we'll talk about that a little bit. And they live in this area, it's the interstitial area. It's called the space of Disse, D-I-S-S-E. All right? And there aren't very many, but when there's liver injury, they multiply very rapidly, and you see more and more of them. Okay? Then you also have the liver macrophages, which are also called Kupffer cells. This is the defense mechanism of your body against toxins that the liver sees on a daily basis, particularly bacterial toxins and viral toxins. Okay, and these are very loosely attached to the endothelial cells which form the sinusoids of the liver. Sinusoids is a fancy way of saying blood vessels. All right? And liver are very fenestrated. What is the meaning of the word fenestrated?
Window seat.
Okay, the window part of it is right. So what happens in a window? Things can go in and out pretty quickly, right? So fenestrated means it's really loosely formed, so things can go in and out of the sinusoids very quickly. And one of the features of Kupffer cells is that they're very loosely adhering cells. So they are on the sinusoids, but they can float in and out of it and attach and even go through these really, really quickly. So our liver is highly, highly fenestrated. And the reason is that all this nutrient-rich blood, and in many cases toxin-rich blood, comes to the liver. The liver needs to filter these in and out of its system with the hepatocytes as quickly as possible. So the liver is a highly fenestrated organ. There's very quick exchange of gases, very quick exchange of materials. And that's how nutrients are absorbed by the liver, processed by the liver, ditto with toxins and also drugs. Okay? There's also a fancy word for bile ducts, bile ductules. It's called like canals of herring, like the herring fish. Some of these classical terms, thankfully, are being taken out of textbooks, as they should be. That was the way when people, if you discovered something, your name got attached to it. Sphincter of Oddi, canal of herring, so on and so forth. So, all right. And last thing to point out, you will see that oxygenated and deoxygenated blood is mixing here, right, in the sinusoids, as I pointed out previously. Okay. One last thing, the interstitial space, where you see the stellate cells. This space also gets a lot of secretion from the cells. So the cells, the hepatocytes, have a major secretory phenotype. So lymph is one of those things that hepatocytes make, and it's secreted into the interstitial place, and then they drain into the lymph nodes. So that was probably the most important slide of the lecture. I mean, I really want you guys to understand how that works. There might be something on the test related to that. Okay? So, fair warning. This is the same look at a liver lobule but from a different angle. The only reason I want to show this to you is because I want to talk about zone 1, zone 2, and zone 3 hepatocytes. So hepatocytes are broken down into 3 zones, right? So the zone that is closest to your portal triad is called zone 1 hepatocytes. Okay? Those that are sort of in the middle are called zone 2 hepatocytes, and then the ones that are closest to your central vein is called zone 3 hepatocytes. And these hepatocytes each have their own characteristics, and that's why it's important. Okay? So the zone 1 is called the periportal zone, and this is the zone that is affected first by viral hepatitis and also ingested toxins. Okay? So this makes sense because deoxygenated blood is coming from where?
GI system. The
Yes. And what does that have?
waste products, toxins.
Bacterial products, viruses, and all that. So the hepatocytes that see this blood are all in zone 1. Right? So it comes out and the hepatocyte sees them. By the way, this is a beautiful diagram of the bilayer of the hepatocytes. All right? So zone 1, hepatocytes are very vulnerable to viral attacks and also bacterial attacks by bacterial toxins and also drugs of abuse. Intermediate zone, right? Not a whole lot of information about them because they're not that interesting. And then you have zone 3 hepatocytes. This is also called the centrilobular zone. And this is the zone that is affected by ischemia. What is ischemia?
Decreased blood supply.
Come on, guys, help me out here.
Decrease oxygen.
Decrease oxygen, right. Okay, so why do you think zone 3 hepatocytes are most susceptible to ischemia? It's logical.
Blood
Who
supply.
hasn't answered? No, you haven't. How about you? What's your name, dear?
Kayla.
Okay.
I don't know, because the central vein is the nearest.
Okay, so that's the right answer because the central vein has deoxygenated blood, right? So everything is draining out and going to your venous return, right? So here, you're seeing more oxygenated blood. Here, there's already very little oxygen. So these are the hepatocytes that are almost always most susceptible to ischemia. All right? These are also the hepatocytes that have the most of the P450 enzymes, and you will learn about P450s in the later part of this lecture. And this is also most sensitive to metabolic toxins, and it is the site of alcoholic hepatitis. All right? Any questions so far? Okay, I can answer your question. You have to email me. No, I'm just kidding.
So we know logically why zone 3 is the zone for ischemia, more vulnerable, but why it's for P450 and for the alcoholic hepatitis. Is there any reason?
Okay, so alcoholic hepatitis is an oxidative damage. Okay, so once again, you have to think around those terms. And what was your other part of the question?
The
Why does it have
P450?
P450? That's the architecture of the liver. That's where you have most of the P450s localized. Okay, that's a good question. I don't know why the P450s are concentrated in the zone 3, but that's where they are. So mostly, zone 1, zone 2 have P452. Your epithelial cells that water your gut also have P450s. But if you were to go ounce for ounce, most of the P450s are in zone 3. All right. So liver function. I just want to touch on it because the liver does so many different things that if I wanted to go through all of it, I could probably make a whole class out of it. All right, 10, 12 lectures. So let's talk about the main liver functions. So the first one that you already would have guessed is digestion. And the liver produces bile, which aids digestion. So bile is really a mixture of water, bile salts, which are produced from cholesterol, and the pigment bilirubin. Okay? Bile salts are 24 carbon water-soluble products, which come from cholesterol, as I told you. So as the red blood cells in our body start disintegrating, the hemoglobin is degraded or broken down into 3 different parts. So one part is the protein part of it, called the globin. Okay? Then iron. Iron is actually very useful to the body for many different enzymes. Okay? And the iron is conserved and taken out for later use. And then you have heme. So the heme is usually broken down into biliverdin, a green pigment, which is then very rapidly reduced to bilirubin, which is the orange-yellow pigment and gives bile its distinctive color. And all these processes take place in the reticuloendothelial cells of the liver, spleen, and bone marrow. So essentially, macrophage-like cells. Okay? And then the bilirubin is then transported to the liver, when it reacts and is conjugated to this more soluble form called glucuronic acid. So bilirubin is conjugated to glucuronic acid, and this more soluble form of bilirubin is then excreted into bile. Okay? There are two big groups of bile salts that are synthesized in the mammalian liver, cholic acid and chenodeoxycholic acid. And each can then be further modified by conjugation to the side chain with taurine or glycine. Okay? Bile is primarily produced by hepatocytes, but it's also produced by cholangiocytes. And I'll talk about that in a later slide. Stored in the gallbladder, and it's released on demand as our body needs it. So the liver is stimulated by vagus nerves. So vagus nerve comes out of your autonomic nervous system. And there are branches all over our body, and this process is controlled by the hormone secretin. And secretin is secreted by enterocytes of your gut. It comes into the liver through, surprise, surprise, portal vein, bringing the hormone into the body. And then stimulation of the gallbladder by the hormone cholecystokinin, CCK. And that's when the gallbladder squeezes. A good homework type question might be to go on your own and look this up and write an essay on it. Okay? One of the big functions of bile is that it can emulsify fats and turn them into micelles. And large fat pieces are broken into small pieces in the process, and they become more manageable. We talked about RBC. Bilirubin gives bile its green color. I already told you that. And okay, bile acids are also very critical for transport and absorption of fat-soluble vitamins, A, D, E, and K. Okay? All right. Go ahead.
People that don't have gallbladders, does it change the digestion function of the liver anyway? I know gallbladder stores bile.
What was your question again?
The removal of the gallbladder.
Okay, so then they're supplemented by all kinds of enzymes to sort of make up for that process. Okay? So in cystic fibrosis, actually, that's what happens. The gallbladder starts losing its function. So if you know anybody with cystic fibrosis, those kids are given all kinds of digestive aids. Okay, so they're always on enzymes like pancreatic amylase, pancreatic, facilitate the process, to compensate for that process. Okay, so very quickly, I don't expect you to remember this, but if you see a cholesterol molecule, you should be able to recognize it. So here's a cholesterol molecule, and then it goes through this whole series of different chemical reactions to make all these conjugates, right? So here's genodeoxycholic acid, where you have a hydroxyl group and a hydrogen group, and then two hydroxyladiol here, it becomes cholic acid. And then they could be, as I said, could be carbon conjugated. So for the sake of time, we're going to move on. Metabolic function of the liver. This is very important. So the liver gets nutrient-rich blood from the gut. It metabolizes carbohydrate, lipids, and proteins into biologically useful materials. But you already knew that, right? For example, just to give you a good example, carbohydrates are metabolized into glucose, and large amounts of glucose are stored in the liver as glycogen, okay? Which are basically long chains of glucose forming a polysaccharide. During periods of starvation, glycogen is then quickly broken down by the liver. I have a slide on that, and released in the liver to produce energy. There are other ways in which glucose can be produced in the liver. So glycerol can be converted into glucose by hepatocytes by gluconeogenesis. Fatty acid metabolism, we'll talk about that in the context of acetic acid, produces energy in the form of ATP. Hepatocytes can also produce lipids like cholesterol, phospholipids, and lipoproteins. Amino acids are broken down in the liver through deamination, producing toxic ammonia that is turned into urea. So urea cycle, maybe remember basic biochemistry, and it's excreted. Amino acids are turned into ATP or converted into glucose through the process of gluconeogenesis, and the liver also stores iron, copper, vitamins A, D, and K, fatty acids, and fatty acids from triglyceride metabolism. So a lot of different things, and obviously we cannot focus on all of these things, so maybe we will focus time per minute on this one function. Okay, so I don't want to go into this in great detail, but diabetes is a big problem in our society, metabolic syndrome and diabetes. And you guys probably know about how glucose is handled, sugars are handled by our body, right? So there are two different ways. So when you have high blood sugar, you secrete insulin, the pancreatic beta cells, and then the insulin is secreted. It helps facilitate the uptake of sugar into all the different organs, skeletal muscles, liver, and some of these things. Now, that's in preprandial state, when your body, in postprandial state, when your body is fed. When your body is fasting, you have the action of this other hormone called glucagon. Glucagon comes into the liver, and it stimulates glucose breakdown. And this is when it raises blood sugar, because now the glucagon is broken up into smaller, not glucagon, but glycogen is broken up into small pieces of glucose again and are secreted out of the body. And this raises blood sugar. This is an electron micrograph of a hepatocyte, and what you should be able to appreciate here is how the glycogen is stored. So these are the polysaccharides, looks like little black dots all over it, right? This is what it looks like. All right? So how does glucagon work? And this is a pretty complicated slide. And once again, there's a little bit of gap in knowledge. This would be a good homework question, that you find out those gaps in knowledge. So I think Aline is an expert on GPCRs, and so I'm not going to talk about GPCRs in great detail. But glucagon signals through a GPCR, G protein coupled receptor. This is a type 2 or a class B GPCR. And class A, class B, class C, class E, class C, there are 5 different classes. All right, and that's because of structural similarity or dissimilarities. And the way it works is, when this is inactive, it's conjugated to the G proteins. G proteins typically have 3 subunits, S or stimulatory subunit and beta gamma, 2 different subunits. All right? What about small G proteins? That's a question from the machine. All right. So what happens is it's usually inactive in its GDP-bound form, but once glucagon binds to its receptor, there's a conformational change, and when that conformational change happens, G GDP is substituted for GDP. So your cell has a lot more GDP than GDP. It's a 10-to-1 ratio. Okay? So as soon as it loses its GDP, a GDP is bound. And at that point, the G alpha subunit separates from the beta gamma subunit. And it starts translocating and starts working on all these different effector molecules. So there are two classical pathways, the phospholipase C and the adenylyl cyclase pathways. There's also a 12-13, G protein 12-13 pathway, which goes to Rho. We won't talk about it here. So when you have this enzyme and it's activated, it works on ATP to make cyclic AMP.
So does the cell have more GTP or more GDP?
It has more GTP.
GTP.
10 times more. Okay? So you start making cyclic AMP by cyclizing ATP, and that's what the adenylyl cyclase does. Okay? And then cyclic AMP in turn goes and activates protein kinase A. Okay, protein kinase A has many different functions. So protein kinase A in turn can go and start phosphorylating some of these proteins and activating here, and also turning on some of these downstream effector pathways such as phosphorylase kinase and phosphorylase. Okay, so phosphorylation happens on one of three amino acids. Either it happens on serine, threonine, or tyrosine. Okay, serine, threonine, or tyrosine residues of a protein. And once this is turned on, what you start getting is glycogenolysis. Your glycogen starts getting broken into smaller pieces of glucose. Now, recent research also suggests that phospholipase C pathway can also do it. In phospholipase C, instead of adenylyl cyclase, what you have is calcium signaling. So you have PIP2, phosphatidyl, I better get this right, phosphatidyl inositol 4,5-biphosphate, okay? And that gets broken into two pieces. This diagram only shows one. This is called IP3. So your homework is to find out what the other piece is. Okay, it's called DHE. So you guys figure that out. Okay, IP3 then has its own receptors on the ER, and then it releases calcium. And then this calcium can go downstream and affect other signaling pathways. So calcium signaling is huge in all types of cells. And then you have reduced glycolysis, reduced glycogenesis. Okay? All right, so this is a rough diagram of how glucagon works in the system. And I think for homework, we're going to ask, there are some steps missing over here. And there's the reference here. Okay, I expect you to learn about this. All right? Okay, liver function detoxification. This is very, very important in the context of drug discovery. So I already told you the liver removes many, many toxins from our and harmful chemicals from our body. And there are 4 phases associated with liver transport, conjugation, and excretion. So it goes from phase 0 to phase 3, and I have no idea why they labeled it, whoever came up with it. Why doesn't it start with phase 1 and go on to phase 4? I don't know. But this is what it is. I didn't make the rules. Okay? So phase 0. This is the basolateral membrane transporters that determine how organic solids enter into the liver. And when the organic solids enter into the liver, it's really the hepatocytes. Okay? So these are on the basal side of the hepatocyte, these specialized proteins that control how organic molecules get into the liver. And here are some examples. Organic anion transporting polypeptides, organic cation transporter, et cetera, et cetera. There's a whole bunch of them. And these basal side, remember, it's facing the blood, so it's facing the sinusoids. Okay? And the apical side is the one that's facing your bile ducts. Okay? So if you think about the liver, and remember it's a bilayer, so if it's a bilayer, okay, this side is your basal side that's facing your sinusoids. And this side is your apical side that's almost in touch with other, and this is the side that's secreting your bile. Okay? Phase 1 are intracellular cytochrome P450 enzymes, and these enzymes are involved in oxidative metabolism mostly of lipid-soluble substrate to facilitate phase 2 reactions. So here are some examples of them, like CYP2E1, CYP3A4, NADPH, LDH reductase, so on and so forth. There's a whole family of them, and I think you guys cover them in literature in lectures down the street. And commonly, but not always, when these reactions happen, a hydroxyl OH group is added to it. Okay? Phase 2 enzymes are enzymes that conjugate many organic compounds with sulfates, glucuronides, glutathione, and acetyl groups to increase polarity and aqueous solubility. Okay, so it takes compounds that have that are non-polar and make them more polar. And when things get more polar, it's easier to secrete those out of the liver and into your urine and other streams for excretion. And you might be familiar with something that people use for Western blot control sometimes, GST, glutathione S transferases, sulfonyl transferases, so on and so forth. These are very important enzymes. And then phase 3, so polar compounds are exported by transporter proteins. Okay, P-glycoprotein is a very famous one. So those who study the blood-brain barrier, or the placental barrier, P-glycoprotein is an ABC transporter. It uses the energy of ATP to transport a wide variety of substrates out of the cells. In cancer, sometimes P-glycoprotein is upregulated. So what happens is cancer cells, they pump out chemotherapeutic agents like doxorubicin, okay? And patients become refractory, so their tumors do not respond to chemotherapy anymore. So things like P-glycoprotein, bile salt export protein, organic solid transporters, et cetera, et cetera. These are the ones that bind up the compounds, secrete them into bile or into systemic circulation for excretion. Okay, the last function, which is often not appreciated, but I think it's vitally important. I like this a lot. So liver produces many important proteins, okay? And these form the components of plasma. And you might be familiar with some of them, prothrombin, fibrinogen. So these are involved in the cascade that leads to clotting of blood. All right, and albumin. Albumin is one of the biggest components of your blood, okay? And they also help maintain tonicity of blood. That wasn't it, was it? Okay. Liver is also an endocrine organ, and it produces hormones. So liver is a function, liver as an organ has both exocrine function, which is bile secretion, and an endocrine function, which is secretion of these important hormones. One of them you might be very familiar with, which is insulin-like growth factor 1, or IGF-1. It also produces prothrombopoietin, which is involved in maturation of megakaryocytes, which is a type of premature blood cells. This, Wake Forest, has many experts in angiotensin signaling, the renin-angiotensin system. And the liver produces angiotensinogen, which is then broken up into angiotensin. Okay? But this is another good homework area because of given the expertise of Wake Forest University, you may want to read up on how the renin-angiotensin system works. And this has many important roles, including cardiometabolic function. Okay? And then the Kupffer cells, we talked about that a little bit. They're the resident macrophages of the liver and play a vital role in combating infections. All right, liver cells. Do we want to take a 1-minute break? No? Stretch? Jumping jacks? Jumping jacks? I do have a Fitbit on it. Anybody else wear a Fitbit? Yes. Okay, liver cells. The liver is home to many different cell types. We are not going to cover all of them. I have highlighted the ones which I feel are important. Now, if your mentor or the lab you're reading through thinks something else is important, by all means read up on it. I have no problem. Okay? But I have my personal bias and personal preference for 4 different types of cells. So we already talked a lot about hepatocytes. We'll talk a little bit about them, some more about them. They're the main parenchymal cells, and they are involved mostly in metabolism. Then you have intrahepatic biliary epithelium, or cholangiocytes. This lines the biliary tree. So remember I showed you the tree-like structure that collects bile and puts it into the gallbladder. Then you have endothelial cells that line your sinusoids and vessels. This is a common architecture that you see throughout the body. We talked about Kupffer cells. They're macrophage-like cells of the liver, so they provide immunity. Then you have NARF cells, and innervation of the liver is much debated and needs to be studied more. I did talk about vagus nerves, how that innervates the liver. Stellate cells, one of my favorites. These are, they're called HSC, hepatic stellate cells. They are a reservoir of vitamin A and fat. And this is a little bit misleading because vitamin A is conjugated to fat and stored in the liver. Okay? And that's the way vitamin A is stored in an inactive form in the stellate cells. Then you have connective tissue cells, including the Glisson's capsule that surrounds the liver. I also showed you another area where you see a lot of connective tissue on a slide. Anybody remember? So Glisson's capsule is an area where you see fibrous tissue, connective tissue. There's also another area in the liver slides I showed you where you see a lot of connective tissue.
Ligaments. The ligaments.
Okay.
Or the triads.
Triads. Bingo. Portal triads. Okay. Then you also have fibroblasts, especially in the injured liver, where cells transdifferentiate stellate cells and turn into myofibroblasts. Liver also has stem cells. I know Wake Forest is doing a lot of work with liver assist devices and things like that using stem cells. The liver can regenerate. We won't talk about this today. And there's this ghost cells of Bower cells, which are stem-like cells. And then you finally have the pid cells, which are highly mobile natural killer cells of the liver. They're mostly cytotoxic, and they protect the liver against viral infections and tumors. Okay? So 70% of your liver cells are hepatocytes. They're the main parenchymal cell of the liver. They're pretty large. They're about 40 microns in diameter. Nice rounded cells. They are polarized cells with the apical surface facing the bile duct tubes and the basolateral surface facing the hepatic sinusoids. I already told you that. As the cells get older, they have polyploidy, and almost 20 to 30% of older hepatocytes will have polyploidy. Mature hepatocytes in the liver do not proliferate. Okay, so they have a very low proliferative index and regenerate in 40 to 50 days. And this is quite controversial, to be honest with you. The reality is, mature hepatocytes are very stable and they don't proliferate. The reason is, when you think about it, you always need, I mean, the liver is seeing so much of the bile, so much of everything, that you cannot afford to have a significant portion of your liver take a break and get replenished. So by design, this is my theory, is that mature hepatocytes have low proliferative index because they ought to be working the whole time. And they have very high metabolic capacity. We talked about all the different things that it does. Fatty liver, steatosis, is a condition of the hepatocytes. And we'll talk about that a little bit, in which you start seeing fat accumulate inside the hepatocytes. And this is a common feature of both alcoholic and non-alcoholic liver diseases. Okay? And steatotic hepatocytes are very vulnerable to other insults. Okay? And we'll talk about that in a second. So let's take a look at hepatocytes. All right. So this is actually work that we haven't published. I told you I work with two receptor systems. One is the cannabinoid receptor system, and one is called the apelin receptor system. You don't have to remember this, by the way. Okay? So, what is this? Who wants to guess?
Central.
Not to somebody else.
Which one? The large circle?
Yes.
A pattern. Portal vein, maybe?
Why would it be a portal then? Okay. Anybody else? Close. No cigar. That's the central vein. And how you know, when you look at something like this, the central vein is, you will see the sinusoids are draining into it. See this? Yes? Your textbook won't teach you this. I am teaching you this. Okay? So, see here. See how it opens up? Remember what the central vein does. It collects blood and takes it out to venous return. So when you look at a slide, and your researcher, when you look at a publication, they just want you to focus on one thing. They might not show you the whole portal triad and the whole acinar structure of the liver. They might just show you something like this. But if you know what you're looking at, you know what you're looking at. In this case, it's the central vein. Okay, these are normal hepatocytes. You see this beautiful structure, sinusoids, and these are mice actually. These are mice hepatocytes. These are mice that have been on a high-fat diet, 60%, the McDonald's diet. 60% of their calories come from fat for 20 weeks. And look what happens to the hepatocytes. And they're full of fat. So this is a stain called an oil red cold stain. Okay? And so you can see two different types of fat accumulation in the liver, the macro and the microvesicular fat accumulation. So macro means they're really big drops that kind of that accumulate in the liver, and it alters the shape of the hepatocyte. And then you have microvesicular, which is really the small little dot. Okay? And this is a liver that has steatosis in it, so as you can see. All right? And what you should also, you might or might not be able to appreciate is that as the shape of the hepatocyte changes, the sinusoids start looking a little bit larger. All right? Okay, so this is, well, these are two compounds that we developed that target the apelin receptor. One is called SRRM247, the other one is called SRRM253. And if you pre-treat mice for 4 weeks, not pre-treat, but treat mice for 4 weeks, starting at 20 weeks, give them this drug at 15 mg per kg twice a day, IP, we can log the process. So now, once again, you know, pretty healthy looking, normal looking liver. All right? This is my only slide of the work I do in the lab. Okay, so alcoholic hepatitis. I told you guys that alcohol abuse is a big problem in Eastern Europe, in Asia, and causes a lot of problems. So, some more tissue features. This is a normal control, and then you get steatosis of the liver as fat accumulates, and it's called alcoholic fatty liver. So instead of oil red or stain, what people do is they conserve or they put their slides in 10% normal buffered, neutral buffered NBF, formalin. Okay, formalin will actually melt away the fat. And what you're left with are these big vacuoles where fat once used to live. So you see these vacuoles. The difference between this slide is this is not a preserved slide. This is a fresh cut, fresh frozen liver. So if you stain it, you can see all the fat with this special stain called oil redo. In this case, they don't use oil redo, but they show you this stuff where you have alcoholic fatty liver. Now, these cells are vulnerable to farther end cells. If you keep drinking more alcohol, you get into hepatitis. Okay? And hepatitis can then go to fibrosis and cirrhosis. So what happens in hepatitis is it's hard to, once again, if you want to become a histologist, a lot of money in that business. So you would have probably missed something like this. This is immune cell infiltration. These are little dots. You see these little dots? These are infiltrating immune cells in the liver. These are inflammatory cells. The other thing I wanted to point out is this little red structure over here with this arrow. This is called a Mallory body. This is an aggregation of cytokeratin into the cell, and this is a classic feature of alcoholic steatohepatitis. Okay? And this is what the liver looks like, if you think about it. It's not that nice brown color, all the fat has taken over, and it looks kind of pale yellow. Questions on this slide?
Can you get cirrhosis just from drinking alcohol, or can it be diet-based as well?
Just by drinking alcohol. Yeah, if you keep drinking alcohol, then the disease doesn't stop here. Okay, then you get fibrosis and cirrhosis. We'll talk about that in a little bit. Okay? Yes, so the answer is yes.
Why is the Mallory body red? Is it stained with something?
Yeah, so it's not a special stain, but it's H&E eosinophilic bodies are red, usually show up as red. So that's too complicated, don't worry about it. All right. Hepatocytes are very critical to drug discovery. For the sake of time, why don't we just skip this one? You guys can read up on it. So we do use hepatocytes a lot in the lab, and so you guys can need to see how we use them. Cholangiocytes. Okay, so cholangiocytes, very briefly, are cells that you find in the intrahepatic and extrahepatic bile ducts. They do a lot of things. They also secrete bile, and they condition the bile by a series of absorption and reabsorption processes. About 10% of the total bile flow in the lab from the liver comes from into the gallbladder, comes from cholangiocytes. Cholangiocytes and gallbladder can be affected by a number of hormones, and I've listed a few of them. So bile ducts and bile secretion, and I mentioned this in passing several times, are affected by many diseases. Okay, biliary fibrosis is fibrosis of the biliary tracts. Biliary artesia, you see this in newborns where the biliary tree and then the gallbladder doesn't have a hole. So you see this in infants and then the bile can come out. Cystic fibrosis could be a good homework assignment. You could read up on why bile secretion is screwed up in cystic fibrosis. Okay. Macrophage-like cells, I already talked a lot about it. Very briefly, this is the largest depot of macrophages in a single organ. They're called liver cells. These are very loosely bound. I already told you that. Primary function is phagocytosis. I already told you that. It releases cytokines. One cool thing, well, not really cool, is that when you have overstimulation of the Kupffer cells by an insult, and we'll talk about that in the context of alcoholic liver disease, they see a lot of bacterial byproducts, right? And then they get activated. And they start secreting inflammatory factors such as inflammatory cytokines, TNF, IL-1, IL-6, IL-8, leukotrienes, reactive oxygen species proteases, and nitric oxide. And this leads to inflammation of the liver. So this in turn brings in other inflammatory cells, and I showed you a slide of hepatitis. So that's when you start seeing liver inflammation. Okay, stellate cells. I do want to spend a few minutes here. So stellate cells initially were called lipocytes or Ito cells because they do contain a little bit of fat, and Ito cells because some guy named Ito discovered them. Okay, they are usually inactive and they're residents of the perisinusoidal space. It's not clear what they do. Most people say that they provide structural support to the sinusoids and the liver. And this is also called the space of disc. So in some literature you might come across this, which is essentially the perisinusoidal space. So that is the space between the sinusoids and the hepatocytes on the basal side. Okay, got it? So stellate cells store vitamin A, which is called retinol, and fats. So usually most of the vitamin A in liver is stored in the stellate cells, conjugated as retinol palmitate. Okay? These are very contractile. These cells are very contractile, and can be activated by certain cytokines. And such as TGF-beta, nitric oxide, endothelin-1, and they can regulate blood flow through the sinusoids. So once again, remember I say they provide structural support. They're contractile. When they expand, stretching, and more stuff can go through. When they do not contract and when they shrink, your layer is a little bit more thick and less things can go through. Now, this is a critical action of hepatic stellate cells. When you have liver injury, now we are getting into the fibrosis realm of liver disease, far more advanced. These undergo transdifferentiation. It starts losing its vitamin A and start proliferating, and it's called myofibroblast-like cells. Okay, and they also start producing a lot of alpha smooth muscle actin. So this is one way in which fibrosis and activation of hepatic stellate cells is determined, at least in experimental animals, is that you look for SMA stain. So the original thought, the original action of HSC is that it is responsible for wound healing. But when you have overstimulation, you have this massive deposition of extracellular matrix proteins such as collagens and laminin in the liver. Now these cells keep proliferating, keep producing all these extracellular matrix proteins, and you have hepatocytes that die off. So you start losing hepatocytes, so your liver function is compromised now. And then you have fibroblasts or myofibroblast-like cells, which can do the same biochemical function that a hepatocyte is capable of doing. So then you start seeing liver scarring and loss of hepatocytes and replacement by fibroblasts, which leads to liver fibrosis. So here's a picture. The liver fibrosis, I think you asked me the question, can be caused by alcohol alone? Yes. It can be caused by other things such as obesity, autoimmune disorders, infection. Multi-step process. Here's a normal liver, nice brown color, and this is a liver with advanced fibrosis. what you can see is that there's a lot of scar tissue, a lot of scarring. Okay, and you have normal liver where you have the stellate cell sitting here happy, and then you have damage, you have infiltration of lymphocytes, that's the inflammatory part of it. You can see that the stellate cells are now starting to multiply, and then they start producing all this yellow discolored cartoon, of course, collagen and all these other things. Your hepatocytes start dying off. So now where you have a nice bilayer here, some of them start dying off. And these are then filled, these spaces are then filled with all these infiltrating cells. Okay? And then what you have is portal hypertension. So your lumen then have increased resistance to blood flow. So the clearance to your liver is no longer efficient. And it's a multi-step process. It usually takes decades sometimes. With alcohol, at least it does. You have fibrosis. First you have the steatosis, then you have hepatitis, inflammation. Then you have fibrosis where you have compromised liver function, and then cirrhosis. That's when the liver fails. And then you have an increased people that have fibrosis also have increased risk of liver cancer. Okay, so what does fibrosis look like? So this is what fibrosis actually looks like. So this is a controlled tissue. This is a stain. It's a special stain. It's called Sirius red stain. S-I-R-I-U-S. And what you see here is severe fibrosis. So if you look here versus here, you will see that a lot of the hepatocyte structure is completely messed up. And then cirrhosis is completely messed up. It doesn't look like these nice sheets, right? And what you see here are the portal triads. Here's a lobule. This is called a bridging fibrosis. Bridge because the fibrosis bridges one portal triad to the other portal triad. Okay? Budding doctors, you will hear the word bridging fibrosis in your career many times. This is one of these. And here is cirrhosis of the liver. Okay? All right, so for the sake of time, we have 10 minutes. I was hoping to be done in an hour. I'm going to let you guys read this through on your own time. I'm going to go through this. But there are 11 million men and 5 million women in the United States alone that are called chronic alcoholics. It's a big problem. Okay? And this slide is actually, let's forget about that. So of these, 90% of the patients will have fatty liver. And then if somebody says, well, you're drinking too much, maybe you should cut back. Well, abstinence will resolve fatty liver. But if you keep drinking, about half of those patients will then move on and have hepatitis. And once you get hepatitis, one of the two hepatitis cases cannot be resolved. There is really no drug for alcoholic hepatitis. The standard treatment is steroids. Okay? And steroids don't really work very well. Corticosteroids don't really work well. There's no real therapy for hepatitis, alcoholic hepatitis. And then one of those two patients will eventually, even if they stop taking alcohol, will progress on to liver fibrosis. So it's a big problem. All right? And there's no effective cure. And then I have listed some of the things a doctor would typically see. I'm not a doctor, so I'm going to skip over that. Some of the diagnostic criteria that one might use. All right, so very briefly, I'm going to go through this. Once again, you could just make a lecture out of this. So alcohol, let's talk about alcohol as a model liver toxicant. So alcoholic liver disease is a big problem. Alcohol causes liver toxicity. And how does it do it? Well, there are many ways alcohol does liver toxicity, produces liver toxicity. So alcohol is handled in the liver by many different systems. So I have only talked about the oxidative metabolism of alcohol in this slide. Homework could be looking at the non-oxidative metabolism of alcohol. Aline's group actually did some work looking at the products of non-oxidative metabolism of alcohol and looked at fatty acid ethyl esterases. Turns out they can turn on the cannabinoid receptor system in the body. So if you're interested, look up her publications, and this would be a good homework too, is to figure out the non-oxidative metabolism of liver. So in the liver, there are three main systems that will metabolize alcohol. The first one is the catalyst system. So this uses the power of hydrogen peroxide, H2O2, makes water, and produces aldehyde. Okay, so ethanol to acetaldehyde is the first step to second system, and by far the most important system, is the alcohol dehydrogenase system, or ALDH. So this system uses NAD, reduces this to NADH, oxygen molecules are added, not oxygen, but ethanol is oxidized to acetaldehyde. And then the third system is an inducible system, which is a cytochrome P450 system. So this is called a CYP2E1, or the CYP family 2 enzyme E1. Once again, this uses NADPH, an oxidant, and produces NADP and 2H2O. So this is kind of a cool reaction. And this is about 10%, this is less than 10%, and this is about 80%. And what happens in chronic alcoholic is that this system is upregulated. So this system starts compensating for these two other systems. And this is by far the most dangerous system when it comes to production of toxic intermediates. Okay? So when you have CYP2E1 formation using NADPH and oxygen, what you start forming is a gem diol. So CH3CH2OH is alcohol. You add an OH group to it, so that's a diol. And then you have an elimination reaction with addition of water. You get acetaldehyde. Okay? This is what the CYP2E1 system does. And whenever the systems are active, you start forming reactive oxygen species because you're adding all these oxygens, you're reducing oxidizing things, and this could be a toxic process in your body. And then what happens is you take, I really want to go there because I don't want to miss anything here. Okay. And then you have another step. So in this case, you have ALDH being broken down into acetate or acetic acid. So you have ALDH, which is now going to take acetaldehyde and turn it into acetate. Okay? And acetate can become one of two things. Acetate is a great source of energy. So we talk about alcohol having empty calories. So alcohol actually produces 7.1 kilocalories per gram. So it's almost as energy rich as a fat molecule. This is about 9. Sugars are about 4, right? So it's very energy rich, but these are empty calories because they don't really produce any nutrition to your body, don't give your body any nutrition. So you have acetate. So acetate is a big source of energy in alcoholics, but typically malnutrition. Acetate can enter TCA cycle as acetyl coenzyme A. Biochemistry 101, everybody took biochemistry as an undergrad? So acetyl coenzyme A can be produced from a number of different substrates, including acetate, okay? And then it can enter the TCA cycle where it's broken up to produce energy. Now, if you drink a lot of alcohol and you have a lot of acetate, guess what's happening? You produce all this extra energy, and how do we store energy in our body? Fat, yes? So that's why alcoholics have steatosis, all right? Now, depending on who you ask, and I'm going to tell you where my bias lies, some people will tell you this is the most important part. You have all this oxidative metabolism going on, reactive oxygen species being formed, and you have all these insults. And if you go here, you will see some of that. Here's CYP2E1, and you have acetaldehyde, you have hydroxy fatty radicals, and this produces a lot of oxidative stress, and you can see all these free radicals being formed. And your reducing capacity of the liver goes down. So you have GSH here, which is getting turned down. And so now your liver is prone to more injury coming from reactive oxygen species. Okay, so that's one world view. You can get lipid peroxidation, and all these things will put an insult on your hepatocytes. The other side of it is you have metabolic stress. You have empty calories, lots of empty calories which have to be stored somehow, and they do get stored in the liver in the form of triglycerides. Okay? So on the other side, one thing alcohol will do is alcohol will disturb your gut. And the barrier of your gut and the barrier of your liver, barrier of your gut is compromised. So now you have a leakier gut. And so a lot of bacterial products can get through and come to the liver. So while the liver is very efficient at removing bacterial products, it gets overwhelmed because there's a bias now towards moving more and more of those bacterial flavors, bacterial products into your liver. The Kupffer cells start seeing more and more of these, and it gets overwhelmed. So when the immune system of your liver starts getting overwhelmed at the macrophage level, guess what the macrophage is going to do? it's going to start secreting all these other sort of factors to attract other inflammatory proteins. This is very common in wound healing. All right? So now your Kupffer cells, they have this receptor called toll-like receptor 4, which is activated by gut-derived endotoxin lipopolysaccharides. Okay? So TLR4, and there's like 10 of these now. TLR1, 2, 3, 4. TLR2 is activated by flagellin, another bacterial product. And so these are on Kupffer cells. Kupffer cells, they get activated, they get overwhelmed, they start secreting all these inflammatory cytokines, and then that in turn recruits other types of inflammatory cells. So you see polymorphonuclear neutrophils in the liver when you have hepatitis of the liver. Where do they come from? They are not resident cells of the liver. And you also get T cells. T cells get activated. And once this starts happening, liver sinusoids, remember the hepatic stellate cells are right over here, they start seeing some of these. Okay, they start seeing some of these TNFs, and some of these TGFs, and TGF beta cells, and that's when you start getting the next phase, which is fibrosis of the liver. So HSCs get activated, they start making a lot of extracellular matrix collagen, and your hepatocytes start dying off, taken over by fibroblasts, and now you have fibrosis of the liver. All right? So Aline wanted me to stop here because the next few slides are reserved for homework. How about we do more, just for fun? Okay, so who wants to go first? This is a slide of the liver. What do you see? Where's the central vein? Is this the central vein? Or is this the central vein?
That one.
That one. Yes, good. What is this? Portal triad. Good. Okay, so zone 1 hepatocytes here, zone 2 hepatocytes here, zone 3. Where's the portal vein? Which one is the portal vein?
Bottom.
Is this the portal vein?
No.
Yes? That's the portal vein. Where's the portal artery? Not the portal artery, the hepatic artery. Remember, exactly, bingo, you got it. And where's the bile duct?
The above.
Right here. Yes, cuboidal epithelial cell, nice staining. Yes, connective tissue. And now you know the liver. All right, so I'm going to stop here. Any other questions, concerns?